Hi everybody. Last week I started this lecture on antibiotic resistance and I'm going to finish it in this video recording. So I will quickly review what I talked about last time just to make this recording complete. I gave a really brief overview of the history of antibiotics, and then I talked about this really cool experiment that was published a few years ago where they had this rectangular agar plate that they inoculated E. coli on each end of. And in order for the E. coli to grow into the center of this giant plate, they had to evolve de novo resistance to the antibiotic at higher and higher concentrations. When I first talked about this in class last week, I said that it took place over hours or days. I looked at the paper and it turns out that this particular experiment took place over 10 days. So several days of E. coli evolution is what you're looking at here. And when I showed this, I talked about how each one of these lineages that we are seeing expanding into the higher concentrations of antibiotic represent independent, parallel evolutions of resistance to the antibiotic. And someone pointed out, oh, but what if they're transferring plasmids? And that is exactly the topic that I wanted to go into next. In this paper, they actually don't mention plasmids and they don't mention the possibility of transferring genes between populations. I actually don't know if they looked into that or have looked into that since this paper. Maybe one of you want to check and see if there were more papers by this group that looked at that question in particular. But in this one paper, they only talked about individual point mutations that conferred these resistances. So in fact, everything that you see here over 10 days of evolution occurred from independent point mutations in these populations. But I do, in fact, want to talk about why the problem of antibiotic resistance is so difficult because of the fact that microbes can transfer their genes among each other so easily. And to explain that concept, which is perhaps a little bit obvious, but it's so important, I want to make sure everybody gets the idea. I have this little cartoon example. So here are some bacteria, and they are producing an antibiotic. And the way this works is that if you are producing a chemical, then the concentration of that chemical is going to be higher right next to you than it is anywhere else. That's just how the physics works. In general, bacteria don't have missiles or rockets or any kind of mechanism where they can shoot out a chemical away from them. If they're producing a chemical, then it can only be transported by diffusion for the most part. And so they're always going to have more of that chemical near them than anywhere else. And so if you are producing an antibiotic, a chemical that kills bacteria, and you are yourself a bacterium, and many of these antibiotic chemicals that we use in medicine are in fact produced by bacteria. That's how we discovered them. Some of them are also produced by fungi, and then this principle is a little bit different, but actually many of the antibiotics we use are invented and produced by bacteria themselves. So if you are a bacterium and you are producing an antibiotic, you obviously need to evolve resistance to that antibiotic if you don't want to commit suicide, right? So this explains why antibiotic resistance is inherently coupled with antibiotics themselves. As long as you have antibiotics, you will always have resistance to those antibiotics. And this is one reason why, which is that most of the inventors of antibiotics, that is bacterial species, have to involve resistance or they will never evolve the ability to produce the antibiotics in the first place. Again, this is just one aspect of this issue. There are other kinds of antibiotics in which the story would be a bit different, but moving on with this particular story, we have this population of bacteria. They have resistance to their own antibiotic that they are producing. The reason why they produce this antibiotic is to kill their enemies or their competitors, other cells around them that do not have resistance to that antibiotic. And of course, these species in turn now have an incentive. It will be advantageous for them to evolve a resistance to this antibiotic, and one way that this gets started is just a de novo independent point mutation in one cell in one population that confers resistance, so that they're the only ones that will not be wiped out by the antibiotic. They can now have babies and they will take over the population, and now all of the surviving cells now have resistance. And this is the mechanism that was driving the process that we saw and the rectangular agar plate with the E. coli in the experiment that I was just describing. So, but as we already hinted, this is not the only way that resistance can spread in the population. One way this could happen is that for these other species to develop resistance, they could have their own point mutations, but in fact that is not what they have to do, and in reality the way that the resistance spreads is that they just steal the gene, or somehow this gene gets passed on to these other species, so now they have exactly the same mutation that this first species has, and that strategy, that mutation for resistance, gets spread not only within a population of the same species, but even to completely different, unrelated species. And that's really how antibiotic resistance spreads so fast. In the video of the rectangular agar plate experiment, it was already evolving quite fast over the course of several days without transfer, and so with transfer, with sharing of genes, it can happen even faster than was shown in that video. So that's the main point I wanted to show, is that horizontal gene transfer, the sharing of genes, is the reason why antibiotic resistance is such a hard problem. So now I want to talk a little bit about horizontal gene transfer itself as an important mechanism in microbial evolution. When I was in graduate school, there was a fierce debate about whether horizontal gene transfer was even important or not. And so this is a famous figure from 1999, which was showing one version of the tree of life. So we have our 3 domains, and everyone agreed that there were some really remarkable examples of horizontal gene transfer, which are the cyanobacteria becoming chloroplasts in plants, and proteobacteria becoming the mitochondria of all of the eukaryotes. And the question is whether the tree of life was really something like this, where genes are constantly being transferred all over the place, even between different domains of life. And in the early 2000s, this was a big debate, and with the metagenomic sequencing revolution, we now know there is no debate. It in fact happens a lot. There's still some question about whether the tree of life is a total fiction because horizontal gene transfer is so rampant or not. Obviously that's too extreme, but there is some still debate about the degree to which the domains are mixing. But no one debates that horizontal gene transfer happens a lot, all the time, and it's a very important aspect of microbial evolution. So here's where we get into more of the didactic teaching of this topic. There are 3 mechanisms by which horizontal gene transfer occurs, which you should know about. The first one is conjugation, colloquially known as bacterial sex, because it requires actual physical contact between the cells that are transferring the genes. For example, through a structure called a pilus, which is like a little tube that the DNA passes through. This is a common mechanism by which plasmids are shared between cells. Plasmids are usually shared between cells of the same species, but precisely because it requires this physical contact, and the physical contact is unlikely to occur between cells that are totally unrelated to each other. So frequently this bacterial conjugation results in the transfer of plasmids, that is small pieces of DNA that are in the cytoplasm and not in the main genome, not in the main chromosomal genome of the bacterial cell. But occasionally bits of the plasmid can be incorporated into the genome. And in that way, this process actually leads to transfer of genes between whole genomes and not just transfer of plasmids. That is, the plasmids can be incorporated into the genome itself. And this is what this diagram is showing. In this class, I don't really get into the molecular mechanisms because there are so many other classes where you can really dive deep into molecular mechanisms, but here's how it actually works if you want to see that. This is not just a theoretical academic thing. This is real. This, for example, is the process by which MRSA spreads, that is methicillin-resistant Staphylococcus aureus, frequently known as MRSA. It's one of these superbugs, and it's serious. If you get a MRSA infection, it can be extremely difficult or maybe even impossible to treat because it is so resistant to all of the clinically relevant antibiotics. And this spreads through conjugation of conjugation that results in transfer of plasmids. Now, this is a microbial ecology course, so I want to explain why and how horizontal gene transfer occurs from an ecological perspective. And it happens when bacteria are stressed out. This is one of my favorite experiments in this context. Here is a microscope image of some cells that have lots of nutrients. They're happy, they're growing well, and everything is fine. Genes are not being transferred. And they know that because in this experiment, they cleverly tagged cells by color, such that cells would show up as yellow or green if a new gene was transferred into their genome, or at least into their cell. All right. And so what this experiment is showing is that if you starve the nutrients, so they become nutrient stressed, then the first thing they do is actually clump together, which is already pretty interesting. Here they're nicely spread out and distributed through the medium, like a proper laboratory culture. And as soon as they get stressed out, they clump together, they attach to each other, and that makes it much easier for them to transfer genes between cells, just because they're more physically connected to each other. And that's when the cells turn yellow and green, because that's how they designed the experiments, to identify the cells that have transferred genes between each other. And that just really nicely demonstrates that horizontal gene transfer is a stress response on a population level. All right, going back to our three mechanisms of horizontal gene transfer, there was conjugation that we were just talking about. The second one is transduction. Transduction is just a weird word to describe horizontal gene transfer that occurs by viruses. So here's a cell being affected by lots of viruses, and whenever you talk about viruses in an ecological perspective, you always have to cite these gigantic numbers because they are mind-blowing. So there are about 10 million viruses in every milliliter of seawater. And so if you do the math, if you stacked all of the viruses in the ocean end to end, they would not stretch to the end of the solar system, not to the next galaxy, but to the next 60 galaxies. If you stacked all of the viruses in the ocean end to end, I don't even know how to think about that. Another way to put it is if you take some experiments on how frequently viruses infect their hosts in the ocean. And the ocean is always used here, not only because it's huge and it makes it most of the Earth, but it's a well-mixed environment. So if you have information about 1 milliliter of ocean, then you can extrapolate to the rest of the ocean. It's a lot harder to do extrapolations like that in soil or something like that. So anyway, there are about 10 to the 23 viral infections every second in the ocean. Okay, that's a lot. Yeah, it's a big number. That's the only point there. There's a really nice review that goes through some of these numbers here that I recommend, that I'm pretty sure is on Canvas. So those are the two of the mechanisms: conjugation, which is bacterial sex; transduction, which is from viruses; and the third one is transformation. Transformation is kind of like the forgotten mechanism because it's so hard to study, because all it is DNA that somehow manages to get transferred between cells with no known mechanism. It's just the DNA is floating through the environment, and cells just pick up the DNA for some strange reason and incorporate it into their genomes, which is even stranger. And yeah, that's what transformation is. All right, going kind of zooming out about and going back to antibiotic resistance from an environmental and ecological perspective. It's very typical to think about antibiotic resistance as a medical problem and an individual health issue. You've probably thought about antibiotic resistance in terms of I should always take the antibiotics that are prescribed to me, because if I only take some of them, then I might be selecting for resistance in my own body. Or I should not take antibiotics unnecessarily. I should not buy the soap with antibiotics, because then I'm unnecessarily using antibiotics on my own body, which also could lead to resistance in my own body. And those things are all fine, but it's really a systemic problem. It's a system-wide structural issue with our society, and not because there's anything wrong with our society, but just because resistance is inherently an environmental issue. You can't think about it just as an individualized medical problem, and here is one slide, one version of explaining why that is. So It's true that if you get antibiotics, you can develop resistance in your own gut, but then that person, at least when there's not a pandemic, meets other people in his environment and can spread that resistance to other people, especially if he goes to a hospital where it's really easy for resistance to spread because there are so many other selection factors in hospitals that can select for resistance. And perhaps just as importantly, and maybe even more importantly, this is an open research question, it could be that our agricultural and industrial processes are also spreading resistance through the environment. It's not only could be, it definitely does happen. The only question is whether it's more or less important than hospital-based resistance. Because we use tons and tons and tons of antibiotics in agricultural practices, and those antibiotics themselves can just get washed off into the soil and into the water that we drink. And then it can select for resistance in the bacteria themselves, and then the bacteria can be ingested through improperly cooked meat and through feces, which then also gets into the water system and the environment in general. So that's especially because of agricultural practices. Resistance is a system-wide environmental issue. And here's just yet another way to think about this from a review that is more from the perspective of what do we do about it, and from a scientific research perspective, what we do about it is, again, it has to be this coupled environmental clinical issue where we really need to study how genes and microbes are transferring between environmental and clinical settings, because many of the resistant infections that people acquire can be traced back to environmental sources. Very few of the resistance infections that are killing people were acquired in hospitals. That is kind of a famous scenario by which people acquire super scary resistant infections, is they get it from the hospital themselves. We know about that because hospitals are kind of embarrassed about the fact that you can get an infection while you're at the hospital. But in fact, that's not where most resistant infections are acquired. Most of them are acquired in the environment just by living in your normal everyday life. And so we have to study all those other sources of resistance, especially agricultural and industrial sources, if we really want to manage and control environmental antibiotic resistance in general. Here's a really good review on this topic of exactly how these environmental industrial practices lead to resistance. And I think I will end it there. See you next time. Bye-bye.